Hello students, this is video 2 of Gametogenesis and today we'll be covering spermatogenesis in detail. I have divided my video and presentation into 4 sections. First, we'll talk generally about gametogenesis and its types and phases. Then we'll talk about the time and site of spermatogenesis. Then the next section we'll be covering the formation of spermatids, and then in the last section we'll be covering the formation of spermatozoa from those spermatids. So first of all, what is gametogenesis? It is the process of formation of haploid gametes from undifferentiated diploid germ cells in the gonads for sexual reproduction. Students, you have to remember that the gametes are formed by meiosis, so they are haploid. However, the germ cells are diploid and are undifferentiated. Now, gametes are of two types. So, gametogenesis is obviously also of two types. Spermatogenesis, that is the formation of sperms, and oogenesis, that is formation of ova. Even though the gametogenesis is of two types, still both involve 3 important phases. The first phase is called as the proliferative phase or the multiplicative phase, where the germ cells are called as gonia. Now students, you should remember that here I am talking about germ cells, not primordial germ cells, as we discussed about them in the last video. Primordial germ cells are formed when the organism is an embryo, and then they migrate into the gonads and help in the development of the gonads. From those primordial germ cells, new cells arise which are called as the stem cells, which help in the growth of the gonad, and germ cells later on arise from those stem cells. These germ cells are called gonia. They may be called spermatogonia or oogonia. And this stem cell population is produced by mitosis, and it produces more and more cells or gonia again by mitosis. So it is a multiplicative phase or the proliferative phase. The second phase is the growth phase. Some of the gonia enlarge in size by accumulating cytoplasm, and they are now called as the spermatocytes or the oocytes. The rest of the gonia keep on dividing by mitosis itself. The third phase is the maturation phase, where these spermatocytes or oocytes undergo meiosis, and here haploid cells are formed, which then acquire structural and functional characters of the gametes. So these 3 phases are common whether in spermatogenesis or oogenesis. First is the multiplicative phase, where the germ cells keep on dividing by mitosis. Second is the growth phase, where some of the germ cells or gonia enlarge in size and accumulate cytoplasm and are now called as spermatocytes or oocytes. And third is the maturation phase, where the spermatocytes or oocytes undergo meiosis and then change into the gametes, that is the sperms and the ova. Now let's talk specifically about spermatogenesis. First of all, when does spermatogenesis occur? So what is the time of spermatogenesis? In some animals, there is a definite breeding season during which the testes enlarge and spermatogenesis occurs. Between the breeding seasons, the testes are small and no sperm formation is taking place, so it is suspended. However, in man and other domestic animals, spermatogenesis continues throughout the year once sexual maturity is reached. The next question is, what is the site of spermatogenesis? Where does spermatogenesis occur? It takes place in the seminiferous tubules of the testes of the male organism or the hermaphrodite organism. These seminiferous tubules lie embedded in loose connective tissue. The loose connective tissue also contains blood vessels, nerves, and a specialized kind of cells called as interstitial or Leydig cells, which secrete the male sex hormone testosterone. The testes are enclosed themselves by a connective tissue sheath called as the tunica albuginea, which is further covered on the outside by parietal peritoneum. The testes may be lobular or may be divided into smaller areas by septa. Seminiferous tubules are lined by the germinal epithelium on the inside. The germinal epithelium consists of the germ cells or the primary germ cells, which are cuboidal in shape, and tall or columnar somatic cells, which are the Sertoli cells or the nurse cells. Now students, you have to remember, in spermatogenesis, meiosis precedes differentiation. That means meiosis occurs first and then differentiation occurs. Now this is the diagrammatic representation of longitudinal section of mammalian testes. You can see here the testes is covered by a layer of tunica albuginea and on the outside by parietal peritoneum. The testis is divided by these septa, which are called as the interlobular septum, into lobules, and each lobule or section contains coiled structures called as the seminiferous tubules. The seminiferous tubules give rise to the tubulus rectus, which join together to form the retitestis at the mediastinum. So these are the fusions of these tubulus rectus. This retitestis then joins with this structure. This is the epididymis through tubules or ducts, which are called as the vas efferens or the vasa efferentia, singular. They lead into the epididymis, which is divided into three parts: the proximal part, which is the caput epididymis, the middle part, that is the corpus epididymis, and the distal part, that is the caudal epididymis, which further leads into a duct which is called as the vas deferens or the sperm duct. Here you can see the histology in the section of longitudinal section of the mammalian testis. This is the parietal peritoneum, this is the tunica albuginea, these are the septa that divide the testes, and these coiled structures are the seminiferous tubules. If we look at this at higher magnification, you can see these are the seminiferous tubules present in the lobule form, and on more magnifying it more, you can see this is the inside structure of one seminiferous tubule. So because these are in section, they are circular or ellipsoid in form. As you can see, the seminiferous tubules on the inside have a cavity, which is the lumen. Now there are two major stages of spermatogenesis. First is where undifferentiated germ cells give rise to spermatids. So it includes all the three phases that we discussed before, that is the proliferative phase, the growth phase, and the maturation phase. And it involves mitosis and meiosis both. In the proliferative phase there is mitosis, in the growth phase there is growth, and then in the maturation phase there occurs the meiosis. So from the germ cells there is the formation of the spermatids. In the second stage, the spermatids give rise to spermatozoa, and this step is called as the differentiation. As I discussed earlier, meiosis precedes differentiation. Now this is the diagrammatic representation. We'll come back to it. So these are the three parts or three phases for the formation of spermatids. The first is the proliferative phase, where the undifferentiated germ cells present in the seminiferous tubules are called as the spermatogonia or the sperm mother cells. The spermatogonia are continuously dividing with the help of the mitotic divisions. They are present in the germinal epithelium. The spermatogonia are diploid. They have a prominent nucleus and they undergo repeated mitotic divisions so that there is an increase in number, which also helps in increasing the size of the testes during early life of the animal. The second phase occurs on reaching the sexual maturity, where some of the spermatogonia stop dividing by mitosis and they grow in size by accumulating cytoplasm and then replicating the DNA. They are now called as the primary spermatocytes. So from gonia, spermatogonia, they have changed into primary spermatocytes. This growth phase is called as spermatocytogenesis. Spermatocyto, that means formation of the primary spermatocytes. The rest of the spermatogonia continue to divide by mitosis. So they continue to divide in the proliferative phase, whereas some undergo the growth phase. The primary spermatocytes then change into the secondary spermatocytes by the first meiotic division. The homologous chromosomes pair up, form synapses. Then the non-sister chromatids exchange segments of DNA, which is called as crossing over, which introduces variation. And the secondary spermatocytes will be haploid because they have undergone the meiosis. As you know, the first phase of the meiosis is reductional. Now in the second phase of meiosis, which is equational, the secondary spermatocytes will change into the spermatids. The spermatids are spherical cells and they have a centrally located nucleus and have all the major cell organelles. Now going back to the diagram, you can see here the proliferative phase is happening alongside, where the spermatogonia are dividing amongst themselves. Then some of the spermatogonia undergo the growth phase. So growth phase is the interphase G1, the interphase S phase, where the DNA is being duplicated. So even though the cell is diploid, having 2n chromosomes, but the amount of DNA is 4 times, so 4C. Then the cell undergoes the maturation. They form the primary, they are called as the primary spermatocytes. The primary spermatocytes enter into prophase 1, undergo crossing over, then go into metaphase 1, anaphase 1 and telophase 1. The primary spermatocytes change into the secondary spermatocytes. The secondary spermatocytes will have half the number of chromosomes as compared to the primary spermatocytes. So these will be haploid. They undergo further division by meiosis 2. So enter into prophase 2, metaphase 2, anaphase 2 and telophase 2. Ultimately 4 cells are formed, which are called as spermatids, each having haploid number of chromosomes. So this represents the first stage, that is the proliferative phase, the growth phase and the maturation phase. This is a combined diagram which shows where this spermatogenesis is happening in the seminiferous tubules of the testes. These are the successive stages that are taking place. So this is the spermatogonium. These are the primary spermatocytes. These are the secondary spermatocytes. These are the spermatids which are formed and which will undergo the next differentiation to form the sperms. So here it is showing where the mitosis is taking place, meiosis 1 is taking place and meiosis 2 is taking place. Ultimately, differentiation will give rise to the mature sperms. This is the actual photographs of the slides of the cross section of the seminiferous tubules. You will see this is the boundary of one seminiferous tubule, which is covered by a smooth muscle layer, and this is the connective tissue present between the two seminiferous tubules. This connective tissue contains Leydig cells or interstitial cells which secrete testosterone hormone. Now when we look at one seminiferous tubule, you will see at the boundary there is present the germinal epithelium having the spermatogonia or sperm mother cells. Below them are present primary spermatocytes. Below them are present the secondary spermatocytes, and then you have the spermatids. Here you can see the spermatids have started differentiating into sperms. Along with this division of the spermatogenesis that is taking place, there you can also see longitudinal cells, these longitudinal cells, which are the Sertoli cells. These are somatic cells which provide nourishment and support to the developing sperms. Another important aspect is that during the spermatogonial divisions, the cytokinesis is not completed. So here you can see the spermatogonia are dividing by mitosis, but there is no cytokinesis. So they form a continuous mass of cells having a shared cytoplasm, which is called as the syncytium. These cells communicate with each other through this cytoplasm. So this connected cytoplasm is called as the cytoplasmic bridges. There is quite an exchange of ions and molecules taking place along these cytoplasmic bridges. Each cohort or each group of cells, because they are connected, they will undergo similar divisions. So all of these cells will simultaneously enter and change into primary spermatocyte, and these will simultaneously go into the secondary spermatocyte stage, and so on. So they mature synchronously. During this whole time, the spermatocyte is forming products or mRNA, which will later form proteins which are required for the formation of the sperms, that is for the formation of acrosome of the sperm or axoneme or the tail of the sperm. And as we can see, during these divisions, the cells move farther and farther away from the basement membrane. So if here there is a basement membrane, as the division is taking place, they are moving farther and farther away from the basement membrane and going towards the lumen of the seminiferous tubule. And we can see that each type of cell can be present in a particular layer of the tubule. If we go back to the picture, you can see here the cells are moving farther away from the basement membrane. Here is the basement membrane below the smooth muscle, and ultimately sperms will be collected here in the lumen, and the upper layer will all have spermatogonia. Middle layers will have the primary and the secondary spermatocytes. The inner layer will have the spermatids, and which then will transform into the sperms. Now we enter into the next stage of spermatogenesis, that is the formation of spermatozoa from spermatids. The spermatids do not divide further, and in all animals they immediately modify into spermatozoa. This transformation is called as spermateleosis or spermiogenesis. So one spermatogonium gives rise to 4 spermatids, which then modify or differentiate into 4 spermatozoa. So this step is quite significant because the testes must produce millions of sperms simultaneously. During spermiogenesis, the developing sperms keep their heads embedded in the somatic cells, the nerve cells, which are called as the Sertoli cells, so as to draw nourishment from them. There are many changes that take place in spermiogenesis. The changes occur in the nucleus, the Golgi complex, the centriole, the mitochondria, the cytoplasma, and the plasma membrane. So all of these organelles undergo changes in the spermatids so that they can differentiate into sperms or the spermatozoons. This is the figure that shows the complete process of spermiogenesis. So this is a spermatid. Here you can see it is having a normal cell structure with the nucleus, the Golgi apparatus, the centriole, and the mitochondria. And here you can see there is the development of the acrosome vesicle from the Golgi complex. So acrosome has started forming from the Golgi apparatus. Here the centriole has gone towards the edge of the cell and formation of flagellum is taking place. The Golgi apparatus or the remnant separates. Here you can see the nucleus has moved towards one edge and there is assembly of microtubules around the middle part. Ultimately, the cell has undergone elongation. Here is the acrosome, here is the nucleus. The microtubular assembly is there, which forms the tail or the flagellum or the axoneme. The mitochondria have assembled into a mitochondrial sheath, and this will ultimately form the mature spermatozoon having a acrosome, plasma membrane, nucleus in the head region. Then there is the neck region with the proximal and the distal centrioles. Then there is a middle piece with the mitochondria, which leads into the tail region, and the end piece which contains the axoneme. So what are the changes that are taking place in the nucleus? First of all, the nucleus takes the eccentric position, that means moves away from the center. It undergoes elongation and it loses water. So the DNA is condensed inside the nucleus, RNA is also greatly reduced, and the chromatin becomes tightly packed. When the chromatin becomes tightly packed, it is unable to transcribe any kind of RNA, so it becomes transcriptionally inactive. All the histone and the non-histone proteins that are present in the nucleus are replaced by transition proteins, which are further then replaced by protamines. Now protamines will have numerous cysteine residues. Cysteine, as you know, is a amino acid having sulfur, so there is formation of disulfide linkages which helps in the stabilization of such kind of proteins. If any protein synthesis takes place during this time, it is only utilizing the stored mRNA. There is a formation of a parallel array of microtubules around the nucleus, which is called as the manchette. The nucleus assumes a particular shape due to the presence of this manchette and this interaction between the proteins of the DNA. The shape is species specific. So each kind of species in its sperms will have different kinds of shape of the nucleus. The nuclear envelope will lose all its pore complexes. The manchette also disintegrates and the microtubules are also lost later on. So these are all the changes that are taking place in the nucleus. Now what happens in the Golgi complex? First of all, the Golgi complex gathers in front of the nucleus. It forms a proacrosomal granule which is having a membrane around it. Many such granules are formed, and these granules fuse to form the acrosomal granule and the acrosomal vesicle. So these are the acrosomal vesicles having the acrosomal granule. This one fuses with the nucleus and indicates that this is the anterior end of the sperm. The acrosomal vesicle keeps on enlarging. The Golgi apparatus then shifts behind the nucleus and is discarded. And this region of the sperm with the nucleus and the acrosome is called as the head region and is surrounded by the plasma membrane. Now, what are the changes in the centrioles? As you know, there are two centrioles. They shift behind the nucleus and lie one after the other at right angles to one another. The anterior one is called as the proximal centriole, while the posterior one is called as the distal centriole. The proximal centriole is at right angles to the axis of the sperm, while the distal centriole is parallel to the axis of the sperm. And it gives rise to 9+2 axial filament. So, if I have to show it by drawing the diagram, the proximal, suppose this is the axis of the sperm. If we look at the proximal centriole, it will be present in this factor at right angles to the axis of the sperm. And if we look at the distal centriole, this will be present parallel to the axis of the sperm. So they are themselves at right angles to each other, and the proximal one is present at right angles to the axis of the sperm, and the distal one is present parallel to the axis of the sperm. In mammals, the distal centriole helps organize the connecting piece of the neck of the sperm and then disappears. In some mammals, even the proximal centriole disappears. However, in case of sea urchins, both the centrioles are retained. Now, what are the changes that are taking place in the mitochondria? In primitive sperms, the middle piece is simple. So it contains few enlarged mitochondria. They decrease in number and increase in size. However, in mammals, the mitochondria shift behind the centrioles and form the middle piece, and they are associated with outer dense fibers, ODFs. They get arranged in a spiral sheath around the axial filament, and they associate end to end to form a helical row of mitochondria. And the region of the sperm containing the centrioles and the mitochondria is now called as the middle piece. Now, what are the changes that are taking place in the cytoplasm? The cytoplasm is drawn out into a thin layer around the nucleus in the head, around the centrioles, the mitochondria in the middle piece, and around the axial filament in the tail. The cytoplasm that is present around the nucleus, which is called as the perinuclear cytoplasm, does not have any organelles. Much of the cytoplasm is discarded and is called as the residual body, and the residual body is then taken up by the Sertoli cells by phagocytosis. Students, you have to remember, during all of these changes, the sperm has embedded its head inside the Sertoli cells. What are the changes that are taking place in the plasma membrane? The plasma membrane extends because elongation is taking place, and it surrounds the acrosome, the nucleus, the middle piece, and the tail. The plasma membrane also acquires certain proteins which can recognize and bind to egg. So there is specific proteins in the plasma membrane that have special receptors that can recognize the egg and bind to it. These proteins are inserted uniformly, but later on they get localized to specific areas or domains where they become functional. Now let's talk about the gene function during spermiogenesis. Whole of this spermatogenesis is regulated by selected portions of the genome. As I told you that the chromatin condenses during this process, so all the mRNA, all the transcription that is done to form that mRNA is completed beforehand only. This RNA, which is synthesized, is stable and is stored and later on translated into proteins. Now, in case of mouse, this entire developmental process starting from the stem cells to the spermatogonia to the formation of primary and secondary spermatocytes, formation of spermatids, and then later on final formation of the spermatozoon takes 34.5 days, 34 and a half days, where the spermatogonial stage takes 8 days, meiosis lasts 13 days, and the spermiogenesis takes another 13 and a half days. In humans, it takes nearly twice as long to complete. Each day, around 100 million sperms are made in each human testicle, and each ejaculation releases 200 million sperms. The unused sperms are either resorbed or passed out of the body in the urine. During his lifetime, a human male can produce 10 raised to power 12 to 10 raised to power 13 sperms. This is the transmission electron micrograph of a human sperm cell. Here you can see the nucleus, the acrosome. These are the centrioles that are present. This is the mitochondria around it, along with outer dense fibers, the axoneme forming the tail and the end piece. These are the references of the sources that I used for making this video presentation. Thank you.